Hello. For those of you that don't know, I'm Steve Russell, currently head of the Department of Genetics, and worked with Michael for many years. So welcome to everyone that's come to this celebration of Michael's life. People that have travelled some extreme distances to be here. I'm very grateful to see you all here. And this is going to be a celebration of Michael's life. The programme, we're going to start off with some introductions to the key areas of Michael's life, his college, the Department of Genetics, and EMBL-EBI. And then after that, we'll have a series of talks according to the programme that you've all been sent out. I don't want to delay the proceedings any further. If the fire alarm goes, just leg it out the door. But the last thing I want to say is to thank Lottie Grukop, right up at the back there, who all of you will be emailed by. Thank you, Lottie. Organisational skills par excellence, and as my PA, I can tell you that is absolutely required. Also thank particularly Ruth Lovering and Cahir O'Kane and Rachel Drysdale, who have also played a huge part in the organisation of this. So, without further ado, now I've lost my programme. Without further ado, who's speaking first? Professor Anne Ferguson-Smith, current Pro-Vice-Chancellor of the University of Cambridge, and the Arthur Balfour Professor of Genetics.
Hi, everybody. A really, really warm welcome to everybody here today. On behalf of the university, a very warm welcome to Francesca and the family, and to Michael's friends, collaborators, and colleagues, and all who've come here to honour and celebrate a very, very unique character, very special person. And I know that some of you, as Steve said, have travelled from far away, so thanks for being here. I'm a mouse geneticist, and I didn't work or collaborate with Michael, so I'll just say a few words sort of from my perspective, the perspective of being sort of in a sort of peripheral layer of Michael's people onion. I did interact with Michael quite a bit, mostly at parties, mostly at Tim Hunt's parties off the Babraham Road, when I arrived as a postdoc in Cambridge, and subsequently conversations on Tennis Court Road and in various meetings and seminars. So that's where I'm sort of coming from, a sort of classic mouse geneticist, developmental geneticist. And we're going to hear, of course, from people who are much closer to Michael than I was. So for those of us who were educated in mammalian molecular and developmental genetics in the late 70s and 80s, I was both in Glasgow and in the US, comparative approaches and the power of model organisms was at the heart of everything we talked about. And Michael Ashburner's name was synonymous with Drosophila and the University of Cambridge, actually. And so I only really knew Michael by reputation as a famous geneticist. But really, when I arrived in Cambridge and started first interacting with him, he really provided for me the example of how academic interactions at Cambridge can and should be. Somebody coming from the outside, a little bit in awe of the place, and interacting with somebody like Michael, who was so human, so non-hierarchical. He engaged with everyone, and he made me feel that here in Cambridge, everyone was valued if they had interesting things to say and do, everyone. And as yet another Glaswegian, finding myself in a rather insular and perhaps somewhat elitist environment, Michael's very creative use of words really felt like home to me. It was very refreshing and down to earth, and really set the tone and told me that here in this place, one could and should be oneself. And for that, I am immensely grateful. Thinking more about the genetics department, I was in the physiology development and neuroscience department across the car park before I became head of department and Balfour chair in genetics, but I always wanted to be in genetics. it was my job. I looked at it from afar, and it was because of the wonderful work that was going on there, the ethos and the values that people like Michael espoused there, the fact that it was feisty and challenging, full of challenging debates, but was surrounded by excellence, uncompromising in the robustness of the genetics, and that Michael really espoused that. So that was one of the reasons that made me want to be part of that family and to have my home there. Michael had always seemed very selective about leadership responsibilities, and for example, he chose never to be head of the Department of Genetics, which I was always sort of puzzled about. But actually, I think that now that Stephen is in the role, we have actually many of Michael's values channeled through Steve, scientific son of Michael, in terms of the commitment to open access, the values, the kindness and generosity of spirit that Michael espoused, and that really is evident in the department. So that's, I think, another important legacy of what Michael leaves. We'll hear more of his leadership from others, but I do want to note that from 1997 to 2000, Michael was a much valued president of the Genetics Society, a position that I currently hold in my copious spare time. And of course, Michael won many awards, but in 2006, he was the recipient of the Genetics Society Medal, which is awarded for outstanding contributions to genetics by an active researcher. And he was active for a very, very long time. The Genetics Society extends its condolences to Francesca and the family, and remember Michael with great, great fondness. Michael the visionary, again, we're going to hear much more of that from others, I'm sure. But our relatively young discipline of genetics has been on an interesting journey, I think, these past 3 decades. It's moved from, in Cambridge at least, from being, I think, a second-class citizen of the natural scientist tripos to touching everything in contemporary life sciences, with the department really being a champion of the discipline within the wider community. And as the genomic era unfolded, and I was in the States when this was happening and watched it, that whole international landscape, many academic departments around the world closed or merged their genetics departments. And I believe that here in the UK, Cambridge remains the only pure genetics department left, although that's not true in the US. But here in Cambridge, and indeed nationally and internationally, Michael really led and held safe this precarious journey between genetics and genome biology that not every institution in the landscape was, I think, getting right. And Michael really drove a vision that made that transition, or that connection, that seamless connection between the study of inheritance and the study of genomes. And he did that through recognizing way, way ahead of his time about the importance of managing big data, data interrogation, bioinformatics, and along with the international collaborators, many of whom are in this room, he prepared the foundation, I'd say, for genome biology today. We'll hear much more of this from others, but this lasting scientific legacy is one that we in Cambridge, as you know, are now embedded in. And we have been extremely fortunate that through Michael, Cambridge has had more than just a seat at the table. So Michael placed Cambridge on the map, I'd say. Not only a brilliant geneticist, but he implemented a vision that provided a foundation for computational approaches to genetics and genomics that underpin contemporary genome biology in its most deep and widest sense. But mostly, he was an inspiration, a role model, a unique and very special individual who, at least in my case, has shown by example how best to try and be an excellent, intuitive, and kind scientist. I think that's all I want to say. I want to thank everybody for coming today, and I look forward to continued interactions with you during the course of the event. Thank you.
Next up, we have Rolf Apweiler, who's currently co-director of the EMBL-EBI. Rolf.
Yeah, also from me, on behalf of EMBL, the European Molecular Biology Laboratory, a welcome, especially of course to Francesca and Isabel. It's great that you are here. You will hear, hopefully, not only the old stories, but also some new pearls of wisdom Michael left for all of us. And I really don't want to talk very long here. I mean, compared to some of you, I knew Michael only a short while, only since 1994. But I can tell you that he was definitely one of my really important mentors. Another one is also here, Graham Cameron, and he will later on talk much more about the importance of Michael in bringing the European Bioinformatics Institute here to Cambridge, actually to Hingston, so nearly Essex, but technically it's still Cambridge here. And Michael played an important role in the life of EMBL already earlier. As for those of you who don't know, EMBL is an international research organization with 6 sites in 5 countries, and we are one of the big ones now here in the UK, and that's really thanks to the vision of Graham, who thought how to set up the EBI as part of EMBL, and Michael to bring it over. And with that, I don't want to say anything more about that, because you will hear all of that from Graham. I only can say his time at EBI was challenging for setting up the EBI, and he and Graham did an incredible job. Getting involved early on through Michael into the gene ontology activities was a pleasure, and his general spirit how to help us to make sure that data is really open and science is open and available to everyone, and that we get the most out of this work, because our work, well, I think I speak for most of us here, our work is paid by the taxpayers, and they don't give us the money to foster our individual careers. They do it because they want to have benefits for society. They may not be immediate and direct, but they will be. And Michael understood that and pressed a lot into this direction. You will hear a lot more about that from others. I really want to stop here and leave the details to the speakers who will come. But say again, thank you all for coming, and I think we will have a very, very pleasant day. Also with a lot of fun, because that's also what Michael always wanted. I'm pretty sure he wants that we have this as a fun event, remembering him, and not a somber event. Thank you.
And finally, for our short introductions, Ken. Ooh. I am not Ken Siddle. Fortunately, we have a fellow of Churchill College here. Professor Cahir O'Kane, former head of the Department of Genetics. He'll say a few words on behalf of the college.
Okay, I don't have anything prepared, so it'll be completely spontaneous. And Michael, as I heard about his science, he was a champion of open science before it had its name. And he was also a member of Churchill, I think from undergraduate, postgraduate, and as a fellow for many years as well. And it's a pity Ken isn't here because Ken's memory of him in college goes a lot further back than mine. But going furthest back, first thing I'll mention is that during the reception, which will be in the Jock Colville Hall, which is just around the corner in the same building, you can go up the stairs in smaller groups to the archive centre, where Michael donated some of his campaigning material from mostly the 1960s to the archive centre, and that includes things like his political activity in the Labour Party, campaign against nuclear disarmament, some reports of the court cases where he was in front of a judge and yes, those days protests weren't seen kindly either, and there's I'll acknowledge Alan Packwood from the director of the Archive Centre, who's put that together. In later times, I think Michael, consistent with his belief in open science, he also had a strong dislike of autocrats and dictatorships, and that even extended to, I think, one of the last times when I remember him being active in college. I think it was about a decade ago, when there was a proposal to set up what was called a Nazarbayev scholarship, and from Kazakhstan, and Michael came to governing body and spoke very strongly against it. So I think that's, if I had time to prepare, I could have said a lot more than that, but time is going on anyway. And just encourage you, if possible, have a look up at the Archive Centre during the reception. And I look forward to the rest of the afternoon. Okay, and thank you very much.
Thank you, Cahir, for that emergency stand-in. If you do go up to the Archive Centre, you will find out, which I was horrified to discover this morning, that Michael once represented the college on University Challenge, which is hard. There's a photograph. It's like something out of the young ones. We'll move on to the kind of more structured reminiscences of Michael's many and varied contributions to science now, and there could be no better person to kick us off than Gerry Rubin, who is currently director of the Janelia Farm Research Campus, who worked very closely with Michael during the era where the Drosophila genome was being sequenced. So it's a great pleasure to welcome Gerry Rubin. For those of you on Zoom, if you want to ask any questions, type them into the chat, and I'll look at the chat at the end of Gerry's talk. If you want a laser pointer, it's up to you. It doesn't move the slides, but you can have it to point.
Good. So I'm going to talk about Michael's role in the Genome Project. This is really what generally happened about 25 years ago. But I first met Michael much earlier than that. I met him in 1971 when I was a first-year graduate student at the LMB, and he used to come over and visit the LMB occasionally. So the Drosophila genome, as most of you know, is a small genome, and because of this, it was selected as one of the key organisms to have its genome sequenced. So those of you old enough to remember back the situation in the late 1980s, there was a big debate in the scientific community whether it was worth sequencing any genomes. Probably 60 or 70% of the people in the community were dead set against it. They thought it was a waste of time. You would get a sequence of A, G, C's, and T's. You wouldn't have any idea what to do with it. We couldn't even interpret the sequences of small genes that we already had, and this shouldn't get done. But the NIH decided to go forward, and they picked wisely organisms where there's some biological payoff. So they picked E. coli, C. elegans, Drosophila, mouse, and humans. But of course, Drosophila genome had been studied for a long time. These are probably the first genome workers, Michael's predecessors in this field. Thomas Hunt Morgan and Bridges, and this was what a genome lab looked like in 1926, 100 years ago. This was Calvin Bridges in his fly room, and yet he was able to make the first physical map of a genome. This is a map that shows a portion of the polytene chromosomes lined up with the genetic map. And they were able to do that because the polytene chromosomes, the banding structure, which we're going to hear a lot more about Michael's use of banding structure and polytene chromosomes to study dynamics of gene expression in a later talk. But it was because the polyethylene chromosomes allowed you to map genes onto them by looking at chromosomal rearrangements and deletions, it provided Drosophila workers with an unambiguously correct physical map, which was very important. No other organism had this. Okay, going forward a little bit further till in 1974, in Dave Hogness's lab, this is just after recombinant DNA, methods were developed in 73, cloned the first Drosophila genes. And this shows, again, using those polyethylene chromosomes where a given clone gene was mapped where it came from on the genome in this classic paper. Now, this is another place where I sort of dovetailed with Michael because I joined Dave Hogness's lab as a postdoc in the 1974, a few months after this, and my job was to make the first big enough Drosophila library to cover the genome. But Michael had been a good friend of Dave Hogness and interacted with him extensively. In fact, his book, this monograph, which if you haven't read it, I strongly recommend it. It's a quick read. Michael comes through loud and clear on every page, but he dedicated this to Dave Hogness, and he, as he writes in this an oral history. There's a lot of good oral history of Michael, conversations with Michael on the Cold Spring Harbor archives. I recommend you listen to him. You want to hear Michael's voice again. It's a good way to do that. So you can see that the relationship between Michael and Dave Hogness went back a long way. So I was sort of interacting with Michael through those years. And Dave was a sort of a visionary about recombinant DNA and cloning, and he wrote a grant request in 1972. There's a portion of this grant request which talks about things which all came to pass of making chromosome maps by assembling overlaps between adjacent sequences and breaking them down. He even predicted the use of bats. So this was a grant that he sent to me so I could write my postdoctoral fellowship proposal. So this was the state of the field back at that point, and then people began to actually put this to work and do assembled regions of the chromosome, and this is the first positional cloning of an interesting gene of bithorax from a 1980 poster. So now we're going to move on, fast forward a little bit to the Drosophila genome sequence. So as I mentioned, Drosophila was picked as an organism to sequence, but no one in the Drosophila field virtually thought this was a good idea. And actually, when the NIH administrators went to the National Drosophila meeting in the US to try to convince people to apply for grants to do this, they said they feared for their life that they were going to be, because people felt, why are you taking our grant money away to do this giant project which is useless? So it wasn't very popular, but a few of us realized that it was going to be necessary and knew enough about how the grant system worked to know that if the Drosophila genome wasn't sequenced, that money wasn't going to Drosophila researchers who would go to sequence the mouse genome or something else. So we set out to do a project in Berkeley, and you're going to hear some more about this from Suzanna Lewis and others, to sequence the genome. And there was a corresponding European project, which Michael and David Glover and Fotis Kafatos and others were involved in, to also participate in this. And we divided it up so they got the X chromosome, and in Berkeley we got the autosomes. We had more money, so we got a bigger part of the genome. And this was all going along in its slow but steady pace when the world of genomics was set on its head by the formation of this company called Celera Genomics by Craig Venter, who announced he was going to sequence all the genomes by a new methodology using whole genome shotgun on the entire genome. So whole genome shotgun had been used on small clones, and it was a standard method, but no one felt it would work on these larger things. And he said, no, this is what we're going to do, and we're going to do it with private money. And this was very controversial. And in 2000, when he announced this, Craig took me aside at the Cold Spring Harbor meeting and said, I would like to do the Drosophila genome just as a test case and I'd like to do it in collaboration with you. And to me, it was obvious, once he said he would put all the data in the public databases, was an obvious thing to do, because it would get the sequence done a couple of years earlier, and it would test this new technology. And I never really wanted to sequence the Drosophila genome. I'm always sort of doing it like as a public service, more or less. So I was hoping I can get 2 years of my life back. So we agreed to do this. That decision was also not popular, and the European collaborators were not thrilled about this. So this is the first case where I would say that Michael was my better angel, and Michael negotiated and brought everyone together and explained to his European collaborators why this is the right way to go, and we got everyone to work together to do this. And there's a number of books that were written about the Genome Project, and of course, Michael's book is just about Drosophila, but there's a book written by John Salston, representing the point of view of the public collaborators, and there's a book written by Jamie Shreve, who was a reporter who was sort of embedded in Celera about the Celera view. And in Drosophila, they basically, all agreed. These books all agree for Drosophila about what actually happened, but Michael's the more entertaining account, as you might imagine. And the surprise to people was it worked extremely well in Drosophila. So most of the government-funded workers in the Sanger Center and the US felt, okay, Greg's going to fail in Drosophila, then he'll go away with his tail between his legs and we'll get rid of him. But instead, it worked extremely well in Drosophila. So this is a collaboration which I think worked well for both sides, an unusual collaboration, because we got the Drosophila sequence very high quality because our combined methods of what we had done between that made the sequence very high quality. We got it done. We didn't have to use any of our grant money to do it. All the data went into public databases. It was all made freely available. And Celera's stock valuation went up $2 billion when the thing was published, because people were betting against that that this would not work. And of course, the money was to be made was through medical research in the human genome. So it was an interesting time. But, and here are some quotes from John Salston's book that sort of just says in someone else's words what I've already told you about. But there's, and then in the end, it was generally regarded as a good thing for Drosophila, maybe not for human, but it created a problem for the human researchers, because now they had to deal with Craig Venter, and it was very controversial about what would happen to the human genome data and things. I'm not really going to talk about that, but it is interesting that even throughout this whole project, this is in John Salston's book, he's describing how Michael was the honest broker and making sure everyone did what they were supposed to do, and held my feet to the fire to make sure I enforced all the agreements we had with Craig. So Michael was there doing this all the time. Then, of course, like I said, it's very controversial. This gives you the tenor of the nature of the interactions between these government leaders, and it was very. It's the only thing I've ever involved in that was this political. I actually had to testify in front of the U.S. Congress about this collaboration. So it was an interesting part of my life, which I'm glad is over. Anyway, all right. But now we're getting this sequence, and we're getting it much, much faster than we ever thought we would get it. And so the question was, how are we going to interpret what the sequence is in a very rapid way? Well, fortunately, we had a model, because there was this study that we did, the Drosophila genome guys in Berkeley, with Michael on this region of the Drosophila genome, which Michael's lab had studied for 10 or 20 years in great detail. So it was probably the best characterized region of any eukaryotic genome as far as how many genes are there, how many can mutate to lethality. And so we said, well, if we sequence this region, we can see where the the mutants are, and we can see what fraction of the open reading frames when mutated had a phenotype, because Michael was convinced he found all the obvious, visible, and lethal phenotypes. And they had mapped hundreds of chromosomal breakpoints in this region to the polytene chromosome map. So it was a really ideal setup. So I remember in the summer when the Drosophila meetings we used to have in Crete, getting together with Michael and said, this is what we ought to do. I'll go back and get my collaborators in Berkeley to sequence this ADH region. And then if we do that, will you work with us to annotate the region? And this led to this sort of wonderful study. With one of the most enjoyable scientific experiments, projects I've been involved in. It was published in 1999. And Michael's work really, again, came back to the old school genetics. It was really like mapping all the genes, the slow stuff, the hard stuff, which then were able to then put together with the genome sequence. And then this was some pictures from that paper. And so the genetic loci, we were able to map them to transcription units here, and then we had transposable elements, insertions that had been mapped, and this went on, for the whole region. And from this, we really showed the power of doing this kind of analysis. And from this region, we answered questions that we didn't have any idea of before. We actually confirmed what people's rough feelings were, that about 1/3 of the genes in Drosophila would mutate to a lethal phenotype. But we also found that the genes that mutate to lethal phenotypes were the ones that were more conserved in evolution, that there was no pressure, they weren't duplicated to protect them. They were expressed at higher levels. So a lot of things we hadn't seen before. But this provided a model. Our view was, this is what we need to do for the entire genome. So this was 3 megabases. The entire genome was 120 megabases. It was 40 times bigger, and we didn't have 40 labs to spend 20 years doing all the pre-genetic work. So we had to try to get them as close as we could. So this is where Michael again came into play to help coordinate this annotation effort. Because we decided, Suzanna Lewis, Michael and I had discussions like, how can we do this? And we said, well, we really need to get like 40 experts from the Drosophila community who know about tyrosine kinases and transcription factors and synaptic vesicle proteins, that knew all these fields, and we needed to get them to come work together for 2 weeks in the same place. We needed to have computational biologists that could help them, and we needed someplace to house this and someplace to pay for it. So paying for it was easy because we could convince Celera that this would make them look good by having the genome being well annotated. That was not a hard sell at all. So they agreed to, and they had expanding their operations, and they had whole floors of cubicle, like office spaces, and they agreed to house us all there. And then we had to get the people. And again, here was Michael was critical because people really distrusted Celera. A lot of the community felt the way about Craig Venter, the way Jim Watson did. So they really didn't want to taint themselves by being involved with this person. And I should say I had a reputation of being pretty rough around the edges and aggressive and other things, so I didn't think people would trust me that way either. And but Michael, everyone trusted Michael. Michael was the honest broker. If Michael said it was okay, it was okay. He gave them what they needed to feel like it's okay for me to participate in this because Michael said it was okay. So he really managed people who would not have accepted to participate in this were willing to accept it when it was Michael trying to convince them. So it wouldn't have happened without the trust people had in Michael. And then we had a group of very talented people. You can hear about Suzanna Lewis, who Jennifer Wortman and Mark Yandell and others, in order to annotate the genome, led to a whole bunch of new software, which led to the gene Ontology, another one of Michael's major achievements. And of course, the NIH people couldn't participate in this because they were like, it would be like sleeping with the enemy. But Michael had all his British friends, and so we had Ewan and Rolf and other people we were able to collaborate with because they were not from the NIH. And we had this wonderful, this is the annotation Jamboree. This is Suzanna Lewis's name for this, after something about Boy Scouts or something, Jamboree. And you can see here's Michael sitting there, and we used to have these meetings. We used to work all day, the biologists. We'd have meetings. Then we'd decide what new software we needed, and then the software developments would come in the evening and work through the night to write the new software. And then we keep doing this, and it went on for 2 weeks. It was extremely, you know, intense. No nights, weekends, nothing off. Everyone was stuck there in suburban Maryland and working in this office building to get this, you know, accomplished. And, you know, most of us were just talking, but some people were working all the time. As you can, this picture might look familiar to many of you. And this led to the publications of the Drosophila genome sequence with the annotations, which came out in March 2000, and then there were subsequent releases and updates of this as time went on and led to the completion of the genome. And I think later this afternoon you can hear about Michael's efforts with Flybase, and here was another case where we had Flybase had this great genetics database, and we were making all this genome database. And when Flybase started, it was way before the idea of sequencing genomes existed, and they just had some note, like, we're not going to deal with DNA sequences, we'll just give you links to the GenBank records. Well, that pretty much became like not a viable strategy once the genome was available, but then there was this issue of, those of you who know Bill Gelbart and know me, that could know that it wouldn't be totally smooth. But again, Michael has said, hey, come on guys, we're doing this for the software community, you guys have to work together, we have to work this out. So I think Michael has always been my better angel, making me a better person and a wonderful friend, and we all miss him. So I'll stop, thanks.
I should check the Zoom chat just to be polite. Oh, good, no questions. God, me next. So, it's a little over 60 years ago that Michael Ashburner joined the Department of Genetics as a final year undergraduate. Thankfully, the zoology department rejected him, and the rest of the world rejoices. He stayed to do his PhD, and then remained in the department, becoming a professor of biology in 1991, an event I remember well. And I think this quote says it all, you know. We have all benefited from Michael's contributions, insights into biology and his encyclopedic knowledge. So his early career obviously was characterized, can be represented as this famous Ashburner model of gene regulation from examining chromosome puffs, and we'll hear some more about this from Ron. His contributions to science were enormous, spanning disciplines from Drosophila biology and genetics, obviously, through to bioinformatics and genome biology, and we're going to hear about these today. There is no question that I would not be standing here today as a professor in the University of Cambridge, to take it from me. It's not the usual career trajectory for hooligans from the west coast of Scotland. Was it not for the incredible support and mentorship that Michael Ashburner has provided me since I joined the department as a postdoc with him just 33 years ago this month, and I have been in the department ever since. But I know this is not just the case for me, it's a case for countless others who'd been through his lab. I was going to put the fly family tree Drosophila family tree, picture of Michael's relationships up there, but it would be like 4-point font and you wouldn't be able to see anything. It is so huge, the number of people that have benefited from Michael's support and mentorship. So as I said, his work really began with his PhD work that he did with Alan Henderson here, who was a lecturer in the department interested in the biology of chromosomes, and he took Michael on as a PhD student. And up in the Archive Centre, you'll be able to see later, these are the 2 volumes of Michael's PhD thesis. Volume 1, 287 pages of text and tabulated data covering the details of, so this is from volume 1, these are tabulated data of puff sizes across development. And where he looked at different Drosophila stocks and Drosophila simulans, made hybrids, and this thesis still reads well. He was such an elegant and precise writer. I learned a lot from him, writing papers with him. I highlight these 2 regions, 75B and 78D, because we're going to talk of them later. The second volume is full of images. A, these beautiful micrographs, and B, his scoring of these puffs. 125 different puff loci he characterized over the course of 12 different larval, pre-pupal, and pupal development stages. Now, I don't know how many of you have tried to make decent chromosome squashes in Drosophila. Never mind be able to read and interpret puffing stages at specific developmental stages. It's fucking hard, because Michael taught me to do it, and I know how difficult it is, and I was never as good as him. But honestly believe that his PhD thesis is an absolute technical tour de force of science. I mean, the lengths he went to control these experiments when making these observations are redolent throughout the thesis, and you can see them in the very detailed methods section where he goes to extraordinary lengths to control the biology in order to make these observations. And of course, the world got to hear about it. So in a 10-year period, you're not be able to read this. In a 10-year period from when he got his PhD, he published 22 papers on chromosome puffing. 16 of these are sole author papers. I do not know of another scientist that has such a, prodigious output as that. The last puffing paper to come out of his lab, actually, was work that I published with Steve Stowers and Dan Garza on the cloning of the 82F puff gene. And Michael said, no, I've not done enough to contribute to having my name in the paper. He did so much more than just this work on puffing, of course. And there's, again, you're not meant to be able to read this, but the work on the ADH region that Gerry mentioned, multiple papers on the genetics and biology of that. Work with Michael Acom doing the genetics of larval proteins. He did early work with Bonner on heat shock proteins, his work on the organization of the genome at a genetic level, including work with David Gubb on autosynaptic elements that still are bamboozling to me to this day. Work on hybrids, which led to a number of important publications on the lethality of hybrids between Drosophila species, and has led to several individuals getting faculty jobs out of what they did. His work on VASA with Paul Lasko, his work on resistance to viruses with DDA Contamine, the work on SOX, which I'll mention a bit later, that come out of the lab. I mean, it is prodigious what he's published, and that truly is a scientific legacy. So let's go back to the Ashburner model, and in a 1973 paper, he'd said the sequential changes that occur in the puffing activity of dipterin polytene chromosomes offer an excellent opportunity for an analysis of the control of gene activity during development. Roll on 17 years and worked from the Hogness lab using this racy new technology of chromosome walking and cloning genes, led to the identification of the gene responsible for the E75 puff. This is one of the early first-induced puffs in the Ashburner model. So as David Seagraves and Hogness' paper says, the steroid hormone activates a genetic regulatory hierarchy manifest by the induction of a specific program of puffs. And the Ashburner had proposed that the transcription of the early genes is directly induced by an Ecdysone receptor, and that the proteins they encode act to replace early gene transcription and induce late gene transcription. So all this bleeding edge molecular biology that went in here essentially led to the conclusion that, yep, observations suggest that E75 is exactly the type of regulatory gene predicted by the Ashburner model, a gene encoding regulatory proteins. And a few months later, work from Carol Tummel and Ken Burtis, also in the Hogness lab, identified another early puff gene, E74, a transcription factor. So 1990 is a momentous year for me because that's the year I joined Michael's lab as a postdoc in October, just not long after that Hogness paper had been published. And Michael had said to me in my interview, come and work in my lab. What would you like me to do, Mike? I'd do whatever you want, but it would be nice if you did something in puff genes. So I joined his long-running MRC program, the genetic and molecular studies of the Drosophila genome. So I set out to clone, along with some colleagues, the E78, which was a so-called early late gene. They were interesting because they required the continuous presence of Ecdysone plus the products of the early gene to be, in order for these puffs to be manifest. So we did the usual. I think we got a chromosome walk from Renato Paro's lab because Polycomb, which he was working on at the time, was just up the road. And along with, I did the usual molecular biology, screening libraries, identifying transcripts, a lot of messy work with far too much radioactivity. Subsequently identified the structure of this gene, two transcripts, which are like E75 members of the nuclear hormone receptor gene family. Along with Adelaide Carpenter, we generated a lot of mutations here from the excision of this P element, plus the usual collection of interesting aberrations that always result from Adelaide's genetic screens, aberrations and inversions, which turned out to be very useful. It turned out that there was no discernible phenotype from this gene. We'd characterized it. We ended up screening 600 lines by southern blotting to identify these chromosomal aberrations, these small deletions here. So that was, somewhat disappointing that we didn't have any phenotypes. But we persisted, and under Michael's tutelage, I became expert in measuring chromosome puffs. So we had our mutations, which we checked out, and we discovered there was a phenotype if you looked at the puffing pattern. So late puffs like 63E and 82F were reduced in size in the E78 mutants, and interestingly, when we overexpressed it, we also saw a similar phenotype. This is a tricky business, and once you have to start doing this, you realize what respect you should have for Michael's technical abilities. This is a filler ocular micrometer, which we would attach to the eyepiece of an aged Zeiss microscope to measure these puff sizes. And the way it worked was, the experiments would be done blind. If for heat shock or the mutants, I would make the squashies. They had to be perfect. Give them to Michael, he would check them out. And if there was a phenotype, then I would make lots more and make all these measurements. So there's 35 different salivary glands here being measured for these things. It is a tricky job, but I'm forever grateful to Michael to teach me how to do this. It was great working with him. Around the same time in the lab, as Gerry has mentioned, there was the European Drosophila Genome Project, which was led by Fotis, and a lot of the work was done in David Glover's lab, Robert Saunders in particular, and they had a unique approach to cloning the Drosophila genome, which was they would take advantage of the physical map that Gerry talked about, the polytene chromosomes, and go in and micro-dissect individual bands. Rather, Robert Saunders would. He would then screen a library, a Cosmid library with those, identify positive clones, there was always lots, try and do some C. elegans style mapping of these by restriction digestion, and then all these clones would have to be checked by in situ hybridization. The clones were all sent to Cambridge, and Tams and Majerus in the lab would spend 5 days a week making chromosome squashes and performing in situ hybridizations, and then generally once a week. And this is what some of the data look like for individual in situ hybridizations. Once a week, or whenever he was in town, Michael would sit down at the microscope and read every single one of these in situ hybridization slides that Tamsin generated, and she'd get an earful if there was any shiny slides which indicated they hadn't been squashed properly. And outside my office at the moment, this is just one of the 30 drawers or so of slides from Michael that he'd scored. Tamsin was doing, a couple of hundred of these a week. Truly prodigious. At the time, I was in the lab, we were doing this puff work. It was taking a long time because we didn't have a phenotype, and the genetics was tricky. But as this was going along, Michael appeared one day at my desk and dropped this Polaroid picture onto my bench and goes, which I interpreted to mean this is from Peter Koopman in Robin Lovell-Badge's lab. He claims it's a Drosophila homologue of the human testis determining factor. Robin Lovell-Badge's lab were doing mouse, human. Peter Goodfellow was doing the human, who a couple of years later became head of department in genetics. But anyway, there was a small tube of DNA, and well, you can't really read this. Essentially, at the highest annealing temperatures with SRY-specific primers, you got abandoned Drosophila. So Michael and I were highly skeptical, fearing that the whole air in Mill Hill was imbued with mouse DNA, and this was just a contamination. But no. Took that little piece of DNA, did the usual, screen a genomic library, screen a cDNA library, single copy gene in the Drosophila genome. I goes, oh, well, that's interesting. And then I did an in situ hybridization and said, Michael, I'm not doing this puff shit anymore. I'm working on this. And it turned out to be Dike, one of the first dominant mutations ever recovered in Drosophila by Calvin Bridges on the 3rd of July, 1915, is when Calvin uncovered this spontaneous mutation, which turns out to be an inversion that results in the ectopic expression of this SOX domain transcription factor. Along with Adelaide, we made a bunch of mutations, and then when I saw the phenotype, I said, definitely not doing any of this, more of this puff shit. I want to work on this. And of course, this gives us another example of Michael's incredible support. So he goes, yeah, well, it's time to renew the MRC program grant, still entitled Genetic and Molecular Studies of the Drosophila Genome. He goes, why don't you write a section on that about Drosophila SOX genes? You can be a co-applicant. And so indeed I did. That was awarded, and along with David Gubb, who was continuing to do stuff on prickle and spiny leg. I cloned another bunch of SOX genes, and was able, the great fortune to be able to supervise 10 PhD students who benefited from all the SOX work that Michael supported. He showed incredible trust and support, and I'm eternally grateful. Again, we're getting towards the end of the century here, and the SOX work is progressing well, and puff stuff is now getting pretty much relegated. And genomics, functional genomics, as it were, is in the rise. So in 1998, there was a call from one of the research councils, the BBSRC, to build genomics infrastructure for model organisms. Now, this is a slide from my PhD thesis where I was looking to isolate differentially expressed genes, and these are sort of predecessors of microarrays, where there's 1,048 lambda clones arrayed out on this square filter, screened with labeled RNA from male or female maggots, and that's my PhD was about identifying sex-specific genes this way. So this, Michael drops the call proposal on my desk and goes, I think we should do something here. Why don't you write something up? So, how many? 85 support letters from virtually every principal investigator running a fly lab in the UK. Michael writes very persuasive letters to get people to send in their support. Then 15 copies of the 100 or so pages that made up this investigating gene function application were couriered off to the BBSRC. £3.5 million, it was a lot of money at the time. And in fact, I'm in this box here that says recognised researcher, because I didn't have a faculty position at the time. And Michael, along with David Glover, who was head of department at the time, sort of used this and goes, look, Russell wrote this grant, £3.5 million, give him a job. And sure enough, they did, which allowed me a few years later to get another £2 million for another 3 years of our functional genomics work. And now I was a principal investigator. I cannot emphasize enough how much Michael's support and trust in leaving me to generate this extremely complex grant application, with a lot of support from him and David Glover and Cahir O'Kane. But nevertheless, you know, this mentorship and guidance was absolutely phenomenal. We set up a genomics facility, we did a lot of genetics, we did a lot of proteomics in collaboration with the biochemistry department, particularly Catherine Lilly, who established the Cambridge Centre for Proteomics. This is a joint facility with the UK Arabidopsis community. We made lots of different microarrays. This is part of the $1 million worth of long oligos and primers that we threw in the dustbin once microarray technology became redundant. We did, with Rob White, we did the first chip array studies in an intact metasome using a genome tiling array, which was the 2.9 megabases of the ADH region. We mapped lots of new transposon insertions and allowed us to take the lead in a European consortium called DROSDEL, where we generated 800 deletions covering the fly genome. John Root was lord of the fly stocks and coordinating this extremely complex problem. And Ed Ryder, a postdoc at the time, led all the sequencing and mapping we did for this. And this expertise and infrastructure allowed us, Catherine Lilly and I, to join a project with Daniel St. Johnson, where we tried to make a whole bunch of protein traps, screening 40 million fly embryos and generating 700 new protein traps. But still, huge amount of infrastructure, and all the time in the background, particularly in the genetics project, Michael was absolutely instrumental. I'm very proud of our microarray facility. We did experiments for over 250 individuals. We were printing thousands of microarrays every year, and then also, adopted more state-of-the-art technology. It was fantastic. And Lisa Meadows, in particular, who now runs the European the Drosophila Stock Center in Vienna, was instrumental here. And Bettina Fischer, who's still in our department, is a technical and computational guru. One last thing is around the end of 2002, a rather quiet-spoken Canadian chap turned up in the department going, can we have a chat? So Michael and I and John Root sat down and talked to Austin Butt, who had this idea that one could develop gene drive technology for controlling mosquito populations, or as some critics at the time said, a frightening proposition that could backfire in the scale of science fiction horror story. This is Austin, and he managed to persuade the Grand Challenges in Global Health and Bill and Melinda Gates Foundation to give us a whole bunch of money to do this. And it's based on this theoretical paper that Austin published in 2002. And the Target Malaria Consortium is still going. It has went from strength to strength. Transgenic mosquitoes carrying a dominant male sterile have been released in Burkina Faso. First ever transgenic insect released in Africa. And the project remains huge and very thriving. Our kind of role, well, the whole concept horrified John Root. So we're not making flies that have a gene drive dominant female sterile or even recessive female sterile going on in my fly lab. You'll wipe out and decimate all the stocks. Michael and I were very enthusiastic, but John was going, no, it will not happen, not in my fly lab. So we built a model system where essentially we set a homing endonuclease target site in the middle of, at the start of GFP, and generated transgenic lines where we could measure whether this enzyme homing endonuclease would cut here and repair by homologous recombination, turning this GFP plus into a GFP minus. And we did. We demonstrated that. We demonstrated we got homing, pretty crap efficiency, but nevertheless we got it. Probably due to the fact that Drosophila males don't do recombination, which is a great thing for genetics, but a bad thing for homing. We also demonstrated we could get homing from ectopic sites. And David Hewen, a postdoc in the lab at the time, was tremendous in leading this work. But throughout all of this project and the project meetings we had, Michael's contributions were enormous, particularly to the genetics. And it's not just about the Drosophila genetics. He had a wide-ranging knowledge of all insect biology, and I think he made some contributions to vector controls consortia in the early 90s, along with Fotis Kafatos. And whenever we had project meetings, of course, Michael would also unfailingly manage to sneak in some time to go and do some bird watching, as remembered very fondly by one of the team members, Sir Charles Godfrey, over in Oxford, and of course John Root, who was his fellow criminal in the bird watching activities, whatever they were doing. It's impossible to discuss Michael without acknowledging his prodigious scholarship, you know. His encyclopedic knowledge of all things Drosophila, and it's virtually unbelievable. The 12 volumes of the genetics and biology of Drosophila that he edited with Nowitzki, Wright, Carson, and Thompson over the years are more than worthy successors to the single volume that Demeric edited in the 1950s. The biology of Drosophila, particularly volume 2D, was particularly useful to me as a graduate student with its reviews in sex determination, dosage compensation, and spermatogenesis. And then the chapters in puffing in 2B when I came to his lab in postdoc. His magnum opus, the laboratory handbook, which Francesca and family have many horrified tales about Michael's single-minded determination to write absolutely everything that could possibly be known at the time about Drosophila into this volume. And of course, the subsequent volumes have still remained, even in this days of instant internet gratification, an excellent source of knowledge. As Graham Cameron will be discussed later, his work in the foundations of computational biology were truly pioneering. In the early 80s, he arranged for the EMBL data libraries to be hosted on the Cambridge mainframe. Here are some of the tapes that are still in my office with the index file here. And he did all this work in his trusty little Mac Plus with its extra disk drive, true power computing at the time. For some reason, this is labeled the John Merriam Memorial Drive, property of UCLA. Never got to the bottom of that. And he used this up until the mid-90s, until he got himself a Sun workstation. Truly remarkable. Michael's spiritual home, as I think Anne mentioned, is the genetics department. When he joined the department, it was located out of town in Milton Road, and then I'm sure you can spot him in a rather typical pose. There's John Thody, then head of department. There's some other, Professor Ingham. People here in the class of 77. In 76, John Fincham managed to get the department relocated downtown into its current location. And at that time, the Fly Lab was located on the ground floor. Then when Peter Goodfellow managed to prize the top floor of our department, which was at that time a teaching laboratory for the biochemistry department out of their cold, not quite dead hands, that was completely refurbished, and the Fly Lab moved upstairs to where the molecular biology lab is, and we had, in 2004, we had John come and open that new Fly Lab. Of course, he was a central figure in Cambridge, and many of you will have seen this picture. It accompanies a review that was published in The Observer in 1988. It's quite interesting. We have Michael Acom, Mike Wilcox, Alfonso Martinez Arias, Michael, Mike Bate, and Peter Lawrence. Too many Mikes, but never mind. A note from this article that Peter's a slight, pleasant-faced man, whereas Michael Acom is recorded as being very tall, thin, and angular. Alfonso and Mike Bate were beer-drinking jokers that lived in the basement of the zoology department, by all accounts. And I don't know what these London-type journals are on. The LMB was considered to be, and I quote, the ugliest building in the city, and whereas my beloved genetics department was noted to be another sensationally ugly block. The bastards. Anyway, tragically, I don't have the bottom half, which I'm trying to track down, because I was shudder to think what their observations on Michael would be in this review. If I find out, I'll post it. And the whole thrust of the article seems to be that there's a whiff of the Nobel Prize in the air. Of course, there was a couple of years later for Yanni, Eric, and Ed for the mysteries of embryonic development. And obviously, in 2002, we, in this very location, held a fabulous 60th birthday symposium, which was a fabulous event, and many of the people here today were at that event as well. Michael was obviously prime pastiche material. So I'm going to give you 4 frames from Beaver Meets Duck. Nick Harding was a graduate student in the department when I left, and this is volume 1, where Michael is teaching his special brand of philosophy, Avoid Ignorance, Prochiality, and Slobbiness. By volume 2, the gnome, as he's known here, had been transmogrified into Old Smoky. I can't think why. Here he is typing away on his analysis of gnome evolution with his trusty little computer there, and stop pissing around out there, do some work or make some coffee. Michael also had a special relationship with others in the department, and I'm not going to say any more about that. You can ask me later. And this is particularly poignant for me, or anyone else that was fortunate enough to be a passenger when Michael was driving somewhere. Heading south for a journey, a meeting of minds. It was a high-speed journey full of danger and adventure. Yes. This is particularly poignant because I believe what Hardin is referring to here is a journey down to the London fly meetings they had at Imperial College. And this would have been late in 1989, and I think that was a London fly meeting where Michael came up and said to me, well, come and work for me. So the rest is history. And obviously, the Old Smoky bit was never far from the minds of the Part 2 students during the annual Christmas pantomime where the students would pastiche members of the department. I can't think where they got the idea for this sort of nonsense from. He was the heart and soul of the genetics department, as Anne has told us, and he taught us so much. Again, I cannot emphasize enough how grateful and privileged I was to have his support, trust, and mentorship over all these years. Coco Chanel said, to have observed that in order to be irreplaceable, one must always be different. And if this is true of anyone, it surely must be true of Michael Ashburner. He will be sorely, sorely missed. Any questions? Good. I'll gladly answer any questions as long as I'm not too rude. Fortunately, Ken has joined us, so we're running ahead of schedule, so we'll get Ken to talk after. It's a privilege to introduce Ron Hill. I first met Ron in 92, I think, when he came over from Australia to teach on an EMBL course that had been organised, and it was a pleasure working with him now, and it's fabulous that Ron has undoubtedly travelled the furthest, all the way from Sydney in Australia. Great privilege to welcome Ron to the stage. Thank you, Ron.
Thank you very much. Yeah. So, in what I'm going to say, I'll draw on a review that we wrote, which was dedicated to the life and contributions of Michael Ashburner, from the Ashburner model to structural biology. So I first encountered Mike through a chap called Mick Callan, who was a lamprey cytogeneticist in St Andrews, and Mick was quite a character. He encouraged me to come from Harvard chemistry department, where I was working on chromatin structure and serotron embryos, to work on newt lamprush chromosomes. Now, it turned out that Mick Callan had been one of the examiners on Mike's PhD thesis just a couple of years before I got there, and so it was inevitable that an introduction we made, and I came to Cambridge for a little while and met Mike, and sort of developed this respect for his incredible work with the salivary chromosomes. It's just quite amazing, the quality of the preparations he made that allowed him to develop this familiarity with the puffing patterns over 125 puffs over some 21 recognizable puffing stages in vivo and in an in vitro system where he incubated the glands in culture and added Ecdysone. And from his familiarity and getting to know these things as friends, he developed the famous Ashburner model. Which served as a great source of inspiration to people in the Hogness lab and other insect molecular biologists to particularly clone some of the elements in his model. The early genes, such as that residing in the 75B puff, which Bill Seagraves made his PhD project, and his PhD thesis was just wonderful. Every chapter was like a paper for Cell. I had the privilege to end up with a copy of it. So walking through that early gene, he found the E75, or that early puff locus, he found the E75 gene, which encoded a nuclear receptor, the typical pattern, transcription, activation, DNA binding, linker and ligand binding domains. And for a while in the Hogness lab, they thought they'd managed to get their hands on the elusive Ecdysone receptor. postulated in the model. But it turned out that it didn't respond to Ecdysone, it didn't bind Ecdysone, and in fact, beyond that, it was this bright red colour, which had no business in the model at all, but it turned out that its ligand in the ligand-binding pocket was a heme group, which led to a protein that responded to nitric oxide. However, it did lead eventually to the Ecdysone receptor via the final chapter in Bill Seagrave's thesis, where he used the DNA binding domain to fish out some 40 related genes in the Drosophila genome, one of which, the hormone receptor protein 23, I think it was, turned out to be the Ecdysone receptor. It also, having the E75 gene and the E75 protein allowed antibodies to be prepared and to check if the protein product of the early gene actually was located on the chromosomes as Michael had predicted it would be, and indeed it was. And this is the very first photograph of Michael's protein P being localized on the salivary chromosomes, and it's a photograph I sent over to Dave Hogness in about 1986, I think, working with him and Bill Seagraves. Today, I'm just going to talk about 3 aspects related to the model. The first is a paper of Michael's in which he gave the prima facie evidence for an Ecdysone receptor. It wasn't just by analogy with what was going on in mammalian systems. He had some important experimental data, which he was quite proud of, in a Febb's Letters paper that's been largely overlooked. He was particularly proud of the fact that Changio was one of the referees for this paper. So we'll talk a little about that paper. Also, I'm going to talk a little about the nature of the molecules in these chromosomes. And finally, I'll talk a little bit about the structural biology, just touching on of the Ecdysone receptor, which my lab's done a lot of work on. Okay. So, it was known that N-ethylmaleimide, a reagent in protein chemistry which reacted with sulfhydryl groups of cysteines, inhibited the receptor activity of estradiol in mammalian systems. So amongst many rather rigorous experiments that Michael did, he incubated glands in his in vitro system with increasing concentrations of N-ethylmaleimide before adding what they call beta-ecdysone in those days, or 20-hydroxyecdysone, or 20E as we'd say now. And the size of the puff he was measuring carefully was 74EF, went down depending upon progressively as the concentration of N-ethylmaleimide was increased in the pre-incubation. So this was consistent with what was known about the mammalian system, but Mike took it one step further. Working in Drosophila, he carried out a sort of competition experiment in which he incubated increasing concentrations of the steroid with the N-ethylmaleimide, and noticed that with increasing concentration of the steroid, the amount of inhibition of formation of 74EF went down. The inhibition went down from 92% down to 60%. And considering all the things that N-ethylmaleimide could be doing in the gland and the cells and all the metabolic proteins and so on, it was typical of his analytical thinking, I guess, that he was able to realize that what he was really looking at was a hypothetical Ecdysone receptor. And he said the action of N-ethylmaleimide is remarkably clear-cut in this paper, this Febb's Letter paper. And it was to interact with this hypothetical receptor protein and block the Ecdysone binding. Now, the simplest explanation for that would be that somewhere on this hypothetical receptor there was a cysteine group in a region close to where the hormone was binding. So you'd get some competition between the N-ethyl going for the cysteine and the hormone binding in that vicinity. And I'll come back to that. So that was one of the things I was going to introduce. And the other is to think a little outside the square on the nature of the molecular biology of the salivary chromosomes. Now, the classical preparations are terrific. They're beautiful. And if you look at the pictures in Mike's thesis, which I recently had the privilege of doing, you can almost feel you could see what's going on a molecular level, but not quite. But you can do a lot with things that involve or reflect primary structure, the base sequence, or amino acid sequence in an epitope. You can localize proteins with by immunofluorescence. But beyond that, we know a lot of things that are important are going on in chromosomes. And they involve higher order structures and macromolecular aggregates or assemblies. And the classical method since 934, when Theophilus Painter introduced it for isolating or making cytological preps, was to squash the gland in various acid fixatives. And if you measure the pH, it's somewhere around, the apparent pH is something around 1.6. And you know what that would do to most proteins, let alone macromolecular assemblies. So I thought, well, why not just isolate the proteins under physiological conditions? And it turns out it's possible. And what I was doing was dissecting the nucleus out, the nucleus out, and then making an incision in the nuclear envelope and withdrawing the chromosomes all under physiological saline conditions. And it's not as easy as it sounds, I can assure you of that. For many reasons, but it's possible, and I persevered. I apparently, about 5 labs in the United States were trying to do that. They all gave up. But working in Australia in isolation, that sometimes has an advantage. And here you see an example. There's the nucleus with the chromosomes in the middle. The nuclear envelope's been torn open. The chromosomes have been taken out, just as a sort of chromatin gel, and they can be unraveled and spread out. This just shows the nucleolus and focusing a little bit higher up, and it's much better preserved than in the squashes where it usually disappears. This is phase contrast, and you don't quite get that sort of resolution you got with the squashes, of course, but it is possible, if you get some practice, to actually spread the chromosomes by micromanipulation with very fine glass needles. And you can see here with the tip of the X chromosome, the resolution's quite comparable with that in a squash. And indeed, if you go to the next level and look in the electron microscope, there's better preservation than in the classical preps. You can see chains of DNA with ribonuclear protein particles growing on them, and even on stalks in some cases. So without the acid fixation, you can still preserve the morphology at the ultrastructural level. And working with these gels, chromosome gels, you can, using antibodies against E75, as Bill Seagraves and I did, localize the protein on this strand of gel, which is 1,000 DNA molecules across. It's just chromatin gel, basically. And as I said, E75 goes back to the early genes consistent with the feedback loop, negative feedback loop in Michael's model, and it appears at other unique loci. that was the very first picture ever taken showing Michael's protein P in situ. And here it's just another example where it's being localized towards the end of the X chromosome. Well, you can essentially do this with the fixed chromosomes using the linear epitopes for the antibodies, the labeled antibodies. So what's the big deal? How would you see a reflection of higher order organization in the chromosomes? Well, this just happened to drop into my lap in a way because there was a big meeting on structures of DNA at Cold Spring Harbor, and there was a lot of interest when it was found that alternating DG-DC copolymers formed crystals, and the structure came out to be a left-handed double helix with quite a different sort of shape. It's just not an entomer of the right-handed DNA, the Watson-Crick structure. And you can see the backbone is sort of a zigzag shape, for example, and they call it zDNA. But the big question was, does it exist not just in the chemistry lab, but in cells? And sure enough, at that Cold Spring Harbor meeting, antibodies against zDNA were found to bind to the polytene chromosomes. So it all was very exciting, and this was known before they got to the meeting, partly why they organized it. However, at the meeting, a paradox emerged, and that was that in Tom Joven's group in Göttingen, the antibodies bound to the bands of the chromosomes, whereas in Alexander Rich's lab at MIT, the antibodies bound to the interbands. And they exchanged antibodies at Aaron Klug's suggestion, and they got the same patterns in their own labs. And no one seemed to be able to work out what was going on, but they sent the antibodies to me to try on the native chromosomes, and the pattern I got was this. There was very low binding, more or less background. But if I touch the native chromosomes for 5 seconds with the acid fixative, immunoreactivity started to appear, and it first appears in the open regions. After 30 seconds of exposure to the acid fixative, there's a massive enhancement of immunoreactivity. So we could get interbands or bands or nothing, and we essentially had the solution to what Aaron Klug, in summing up the Cold Spring Harbor meeting, referred to, I think, as the zDNA paradox, the band-interband paradox. So I submitted it to Nature, and blowed me down. They sent it back, and the editor sent it back, but he knew what was going on. The referees had reviewed the paper, the ones who'd kind of made a bit of a mistake, and so he said the editor sent it back saying he was sending it back reluctantly, which is not all that usual when Nature writes back to you if they're sending it back. But he said, well, I'll do one more experiment and resubmit it. So I was thinking now, what's converting bDNA to zDNA? And thinking about it, the nucleosome, which is a macromolecular assembly, which I thought would be destabilized under acid conditions, has DNA in a left-handed superhelical path on it. And as you know. And if you disrupt the nucleosome, the DNA is freed. There's a whole lot of free energy of deformation in a 20 angstrom wide double helix being wound around a small core, so the whole thing's about 100 angstroms in diameter. And if I calculated, if you release that free energy of deformation, it will turn into a twist eventually, a torsional stress and then a twist. And that would be sufficient to convert bDNA to zDNA. So the experiment I did to send it back was to treat with topoisomerase 1. If I treated with topoisomerase 1 before the acid exposure, we still saw the immunoreactivity. But if I treat it with topoisomerase 1 after treating this material with acid, the Z amino activity completely disappears. So the torsional stress released on disruption of the nucleosomes drives B to Z. And if you get rid of that torsional stress, then the DNA stays as B DNA. And interestingly, over recent years, a new class of anti-cancer drugs called the curaxins has been worked on, and I think it's in some stage of trials now, but it appears to act by destabilizing nucleosomes and causing zDNA to form. So that mechanism is actually happening in vivo, and I wrote some reviews suggesting that it very likely would, and at least zDNA would exist as a transient state, and I wasn't saying the whole thing's an artifact by any means, and that can be associated with transcription and so on. Okay, so the chromosomes. I think there's questions that can be answered by having native chromosomes, and interesting possibilities there. But finally, let me just go over to the structural biology of the ecdysone receptor. We've been isolating ecdysone receptors from a number of insects, particularly pest insects in Australia, like the sheep body louse. And because of the breakthrough that was made with the E75B, to get the ecdysone receptor from Drosophila, we can then use that as a probe to get the ecdysone receptor from many pest insects, and there are now hundreds of them available. This just shows one example. There's two subunits. The ECR subunit and the USP subunit, which are both required, and certainly in higher insects, to form an active receptor. And we've been able to crystallize these. They're very difficult proteins to crystallize. The first one took us 3 years, and in Dino Maras's lab, they crystallized one. It took them 3 years too for the first one. You see the USP subunit and the ECR subunit. Within the ECR subunit, there's a pocket representing transparent grey here, and you can just see ecdysone and ecdysteroid inside that pocket. Now, we're interested in trying to work out why different ligands interact with ecdysone receptors, particularly in different insects. And so we've done quite detailed studies on analysing the amino acid residues that make up the walls of that ligand-binding pocket. And that's another story. But interesting for today, there's just one cysteine residue associated with the walls of the pocket. And there it is. So, could we be looking at the very cysteine that Michael was looking at in 1972 with an ethylmaleimide reaction? And if you do modeling, then you find here's the steroid, here's that cysteine and the sulfur atom, and it's virtually in contact with the steroid and no room for any ethylmaleimide to fit in. So we would postulate that this is the explanation for Mike's data 50 years ago. But there are a lot of other cysteines around, and there could be something else going on. However, we did an experiment which we replaced that cysteine with a serine, so we now have an oxygen atom here. And, okay. In black, you've got the wild data of hormone binding to the wild type cysteine-containing protein. And here is the binding in the absence of any exposure to any ethylmaleimide. But if you expose the protein in vitro, the purified protein, to any ethylmaleimide, the binding drops very dramatically. The hormone binding drops very dramatically. This is at about 10 times the molecular concentration of the receptor. You see it dropping down from there to there. And then if we go to the concentration Mike was using with the glands, another 4 times higher, it drops a little bit more. If we use the protein and purify protein, which has an oxygen atom instead of a sulfur, a serine instead of a cysteine residue, then you'll remember that cysteine is very close to the binding, and the steroid doesn't bind as much, but it still binds very significantly with the serine instead of a cysteine. The difference being that now, if you add increasing amounts of N-ethyl-leamide, there's no significant drop in the hormone binding. So it would appear almost certainly that cysteine is the one that Michael was reacting N-ethyl-leamide with in those experiments so long ago. And it's sort of typical of the rigor of his thinking that he would see right through to effectively be seeing a manifestation of what we're now seeing. And my final slide just harks back to the Ashburner model, and this was just not a guess. It was based on experimental evidence, as was the whole model. You can see here just a depiction of the N-ethyl-leamide trying to get in between that cysteine and the steroid, or if the steroid, if the steroid's there first, the N-ethyl-leamide can't get in, which he also found in another experiment. So, all hail to Michael Ashburner, and I'll end at that point. Thank you very much. Thank you. Thanks, Steve.
had to be, this was in the days when everything had to be compiled on a particular machine. So this was to run on the Mac, that Mac that you showed a picture of. He wanted it to run on his Mac. So we put it together, and at the, let's see, which meeting was it? It was in, all right, I'll get to that later, but just, Richard organized a meeting for everybody who was developing versions of ACDB, organized out at the MRC offices at Addenbrooke's. So I went and attended that meeting. And then I was going to go in and meet Michael for the first time. And it was Cambridge, so it was a little too far to walk to get to the Downing site. I didn't have a car. The bus schedule didn't work, so what do you do in Cambridge? You take a bike. Of course, I didn't have a bike with me, so Richard Durbin lent me his rather quaint bicycle, had steel rod brakes, and also Richard is considerably taller than I am, so so I'm riding this bicycle in. It's a hot day. The traffic was quite heavy. So I'm nervous. And also I can't really stop, because if I stop, I have to lean the bike way over so I can put my foot on the ground. Anyway, when I finally get to the Downing site, I'm red, I'm sweating, I'm just shaking. Michael was so kind. He just got me some water, introduced me to Steve. As we walked through the labs, we get up to his office. And I remember both the paper mache fly that he had sitting on top of the filing cabinet and the letter from the zoology department rejecting him. He was very proud of it. He pointed it out to me. And, and that was it. That was the beginning when we began collaborating for the next 20 years. And so we did get a FlyDB burnt onto a CD. That was the latest and greatest technology at the time, was a little CD disk. And we handed them out at the Atlanta, Georgia Drosophila Conference that year. We shipped out boxes of these CDs. And we were celebrating up in the top floor of the hotel the meeting was held at. And someone came up with a copy of the Drosophila book. And they wanted his autograph. And this is when I realized, oh, Michael's kind of a big deal, because I'd never met anybody who had sort of groupies before. So, because I was not coming from the Drosophila world, I'm more of a software person. And so that was getting to know Michael. The next project was ADH. As Gerry mentioned, we were doing sequencing. We needed a pilot project because, as all those detractors to funding the big genome sequencing projects had said, what good is a bunch of DNA? You need to annotate it. And this was in the early days of gene prediction software. HMMs, hidden Markov models, were the latest technology for predicting the genes. And so we wanted to augment it and basically help debug the gene prediction software by doing visualization. So in Berkeley, it was Seema Misra and I. We sat side by side at the bench, and I would hear her mutter about something. She would never complain. And then I'd sort of slide over whenever I heard her muttering, and I'd say, "What is it, Seema? Show me." And I would add a new feature to the visualization, because at one of these, someone showed from Asilomar, I think it was, on one of the previous slides, like paper taped together. And that's what we had on the wall in front of us, was the ADH on paper, which later was in the publication, as Gerry showed. And the timing, though, is the 8-hour time difference between California and here in Cambridge. And it worked out beautifully, because Michael and John Root would send us their questions and requests. And so we would come in the morning and see a bunch of emails from Michael and John, and then we would send back answers and our own questions, and it just ping-pong back and forth every day, and it just worked out beautifully. And then we didn't publish right away. Since it was the time when gene prediction software was just new and just being developed, and this was an region of the genome that just had been fairly carefully and verified in the lab, annotated. We wanted to give the gene prediction developers the same ingredients we had had to work with and to see how well they could do. And so we organized a little mini competition in 1998 at the ISMB meeting that was being held in Heidelberg. And so everyone had the genome sequence that we had, everyone had the cDNA sequence that we had. And it was really productive. Roderick Guigo took over that sort of same effort and repeated it for a few years afterwards to try to keep on improving the gene prediction software. And and then we published the paper. And one thing Gerry didn't mention, but this gets back to the kindness that several of you have mentioned, was while I had been sitting next to Seema on our bench where we worked, she had been getting bigger and bigger because she was expecting her second baby. And we got the paper published just in time before he was born, and Michael dedicated the paper to her son, Aiden. And it's that generosity of spirit that really reminds me of Michael. So the fly genome. So now we had an idea of how you're going to do annotation. We needed to do it to the entire genome. And as Gerry said, this is when we switched gears entirely, because I'd been building maps up the back sequences, like which back overlaps which back. But now it was going to be whole genome shotgun. Fine, sooner, better, cheaper. So Fly was essentially Celera's pilot. It was, Fly was to human as ADH was to Drosophila. It was their version of getting things going. So one thing about Michael was he was very much open access. He called it open kimono, which is, he was very much that way. It was that brashness, yes, it's going to be full open kimono. And it did get smoothed over, Gerry, but when the first Drosophila page on NCBI was out, Michael saw it, and it was just like, I'm going to quote a few things from the how the fly was won, but the sequence, this is the NCBI Drosophila page. The sequence information provided is for your personal and non-commercial use. You may not copy, distribute, transmit, reproduce, or sell any of this information in whole or in part. Michael went ballistic. And so this really stuck in my memory. He called Gerry, certainly. He called Francis Collins, who, head of NIH. He called David Lippman, head of the NCBI. He was really panicked, and he was going to, Gerry got everything calmed down, but he was going to call everybody who was invited to the jamboree. He was going to call them, say, don't come, it's over. And this is, in Michael's own word, it's off, it's over, it's kaput, finished, done for, dead as a parrot, non-jamboree. He didn't have to send it because Gerry got things straightened out. And so then we shipped off the sequence to Cyrus's company, actually, Neomorphic, where David Culp and Martin Rees ran the gene prediction program, Genie, over it, and we got our 13,600 some odd genes predicted. But then we had all these people coming, 40 people, 13,600 genes. You don't want to just give everybody everything. We wanted to partition them. And of course, Michael, who had been working for Flybase, had developed a controlled vocabulary to describe the function of genes for fly. And he wanted to use that as a way to bin the gene predictions appropriately, so that the people who were interested in olfactory receptors, for one thing, could come and get a set of genes that might hopefully be related to olfactory reception. And so we had people send us the sequences that they were interested in, that they were working on. We collected a few others. Mark Yandel wrote a little Perl program called Love at First Sight, which then we ran over all the genes and compared them, using the evolutionary approach, like what does this look like? Which one is it similar to? So that then we could bin the genes roughly and give, so that when people, these were like party favors when they arrived at the Jamboree, to work with and study in those meetings that we were held every afternoon. And then, Mark and I would work over the night along with others. And so that was getting but that was a proto-Go. So that was a Go that was developed primarily for fly. But the year before, in 1998, Michael had written a white paper that he presented at the 1998 ISMB conference in Montreal. And was not successfully received. This was a bunch of what I would call theoretical computer scientists. They didn't actually want to get software built that was useful. They were going pure algorithmic theories, which is useful, but Michael's approach was a little too pragmatic for them. And Go came in for a lot of criticism then, now, and ongoing. I think we encourage it. Because Michael, in some ways, reminds me of Cyrano de Bergerac, independence and frankness, and he welcomed the criticism, and because it helped improve the Go. And as long as it was doing that and wasn't just, well, look at me instead, I've got a better approach. But as long as it could be helpful, he was open to it. And like Cyrano, he could probably find more problems with what we were doing than any of the external critics. So he was always very open to it. So now we began to bring in other species to apply these, what are the functional, have a common language for describing the function in the genes. Because this is what you need. If you have a Tower of Babel, you're not going to be able to realistically compare results, because are you actually talking about the same thing? So Michael's, compulsion to organize and categorize everything, it was just like, there's more species, there's more genomes, there's more gene predictions, let's go ahead and do it. Every single time with Go that we brought in a new species, revisions were mandatory. I assume this is still true. But even one of the first organisms, which was yeast, along with fly, was like, oh, chitin. Chitin's not in the right place, because chitin for yeast is part of the cell wall, but chitin for Drosophila is more the exterior of the fly. So this kept going, and also, intracellular. When we started working with some parasites that invade the cell, it's like, oh, you have to be careful of what perspective you're using. So that it was always, he loved that. The other thing that came out too, actually, was that we needed other types of ontologies. We needed all the cell types. We needed anatomical cross-species anatomy. And he has consistently, he would welcome and bring in new people. And now he started, I think it was 2002, the Open Biological Ontologies, which is a collection of complementary ontologies that work together. This is still vibrant. There's so much that Michael did that the repercussions continue on and on. Looking back, Michael was usually looking far, far ahead of where most everyone else was. And when we were doing the annotation for Drosophila, and of course it was the evolutionary approach we took to bin those genes, and then we met like a year or so later when Solero was doing human to talk to Paul Thomas about the annotation for human and what would be needed. And Paul Thomas, he said he was just astounded at Michael's encyclopedic knowledge. It was like whenever he showed them the protein family tree, and he would know, be able to dig down into the details. He just didn't recall it. It's like those textbooks, they were all also up in his head. We usually had lunch at a sushi place. Do you remember that? Michael loved uni. And I do remember the live lobster that we got a lesson on some arthropod anatomy over lunch. But a few years later for Go, we were having a meeting in Palo Alto with Mike Cherry at dinner at a sushi place, sketching out on a paper napkin a tool for using a phylogenetic protein tree to annotate Go, so that we actually use Go to describe the functions of ancestral proteins, and then they could be propagated down to extant proteins. Michael has actually helped with the quest for orthologs, which is another thing, Rolf, that you've helped with. And this, I remember sketching it out on a paper napkin, and this tool paint is now used by Go curators to annotate across species so that you can take the experimental results for one organism, put them into this evolutionary context, and draw inferences that way about when did that function arise or when was it lost. And this urge to collect and organize information, I think, permeated his whole life. I know he read, every book in a mystery series, if there was a series of books by a certain author that he liked. He would listen to every opera and the birds. Because all of these research collaborations that he had going involved a lot of traveling. So whenever he went to a new place, he always looked up where are the birding sites. And he would get up at the crack of dawn. And once or twice I got up at the crack of dawn out of curiosity. And it was like, get up, get a cup of coffee, and go out to some birding site to see whistling ducks or something. And I hadn't realized how much risk-taking was involved in birding. Or just riding in a car with Michael, and it's like not just down bumpy dirt roads, but directly across the field, if need be, to get to where you needed to be to see the birds. And, or just getting down on your hands and knees, crawling through the shrubs to find some little bird. Michael was a complete joy to work with. He had a huge impact on my life. And as he said in that email to me, it was always intense. It was fun, and it was productive. And I don't know if I should do this one, but I asked him once how he felt about climate change. He was very sanguine about it. He said, species die. That's what happens. And it's like, as in Ecclesiastics, it's the same destiny ultimately awaits everyone. Righteous or wicked, good or bad. So whatever you do, do it well. And he did. not quite as scientific as some of the others.
We'll have a break now, I think. And return that. Return about 4 o'clock, where we'll kick off with Ken for a couple of 10 minutes on his recollections about Michael at Churchill. Thank you for all the speakers in this morning's session. It's been this afternoon session. It's been fabulous. Thank you very much, everyone.